Welcome to this webinar. And this is a series of webinars in which the Pandemic and Disaster Preparedness Center presents its so-called front-runner projects. And these are projects organized around 5 complex themes that we feel are going to be big challenges for the future. Now, the focus of today is a complex convergence of problems related with climate change and vector-borne diseases that will be introduced by the speakers of today. Now, why this and why here? Well, one of the reasons is because we are good at it in the Netherlands. The Netherlands is being looked at as a country for solutions when it comes to problems related to water and climate, because of our history and track record in that area. But the Netherlands also is a front runner when it comes to expected impact of climate change. So the projected temperature increase is quite considerable in this region. This also has effects on sea level rise, and there's projected impact on aspects of weather and extremes in weather, all of which have an impact on this complex environment. And we are not alone in this. There are other delta regions in the world that not only have the same complex changes to deal with, but also are very densely populated and highly globally connected, just like our Delta region is, in which you can expect to start to see complex problems. And the problem that we are superimposing that is a problem that we see everywhere where there is big change coming, and that is the risk of new infectious disease outbreaks occurring in these systems. The one that is superimposed here today is focusing on infections that can be transmitted through mosquitoes or vectors, and think about diseases like malaria or what we call arbovirus infections, arthropod-borne virus infections, with examples like dengue, yellow fever, Zika, and West Nile virus. So today's consortium that presents itself is really a much bigger consortium that has been discussing this complex combination of factors and what would be good topics to research going forward. The presenters are highlighted in yellow here, but as you can see, this is a collaboration between several institutes with a broad range of expertise, and that's exactly the type of collaborations that we want to see develop with the PDPC. There's also links with national and international initiatives that are important for this discussion. So that's my brief introduction, and let me hand over the floor to the first frontrunner presenter, Julie Pietrzak. I'm not sure if I pronounce your name right here, from the TU Delft. Julie, please take the stage.
And I hope you can hear me. Thank you very much for that introduction. So I'll just start here. So as Marianne mentioned, my name is Julie Pietrzak. I'm a professor of physical oceanography at TU Delft. A lot of this work I present here today is based on work we're doing in salty solutions, and I'm the program leader for that. And it connects very nicely with ongoing work, I believe, with the viruses as well as with groundwater. So what are we looking at here? I like this photograph because you've got a nice image showing basically the problem we have with salt intrusion at Delta. So this is the mouth of the Rotterdam waterway. We have our flood defenses, we have the port, we have industry, and we have agriculture. And all of our deltas worldwide need fresh water. We need it for all of these things, processed water for industry, drinking water, etc. But we have one small problem in our deltas. We've got this salty seawater out the coast. So here's the coast and the salt water. If you imagine this is an experiment at Deltaris, and if you had a partition and you separated fresh and salt, you remove that partition, the salt water moves underneath and the fresh water moves out to sea. So the salt water moves inland and it has an equilibrium position, it moves with the tide, but the drinking water companies know where it is typically now and they place it, freshwater intakes accordingly. But with climate change, we're likely to have some problems that we have to think about with designing our delta. Sorry. So basically, salt intrusion is a growing problem. It is caused by surge at the sea combined with low river discharges. And in 2003, we had a major salt intrusion event that went all the way up towards Gouda, about 60 kilometers inland, and caused problems for drinking water. And we have the problem of climate change. So I present this little movie here to illustrate it. This is a simple, an older version, the water management system, the network model. Now, fresh water, we want water that has a chloride content of about 150 milligrams per liter. Anything that's dark blue is already a problem, and red is really not very good. It's not where we want to be. So what you see here is that same example from 2003, but under extreme climate change. So you see the salt is spreading throughout the whole system. So this is a potential concern, and we're looking at ways to mitigate for the future. In 2018, there was a major drought. Again, there were problems throughout the whole system in trying to figure out how to get water to the western part for agriculture, fresh water, etc. As I mentioned, it's a global problem. So many of our deltas worldwide have issues with salt intrusion that are only going to get worse with time. And thanks to Tom Bogart, I have these slides for you showing the other problem beneath the ground. We have salt intrusion in our deltas and also in the groundwater. Here you can see the fresh water, the brackish water, the salt water beneath the surface, beneath the ground. And we also use this for freshwater resources. And so we really want to minimize salt intrusion in our deltas beneath the ground as well. And here's a little movie from Tom. It's a sequence of images to show the problem. So this is using a cross-section near Amsterdam. And here you see the sea, the dunes, the tulip fields and the polders, and you can see underneath the ground the fresh water, which we need for agriculture. Now, if we look here, this is an example from 1980. You can see quite some fresh water in the required regions. Now you see 2020, still okay. And now you go to 2060, beginning to see problems with the salt intrusion in the groundwater. And by the time we get to 2100, there are quite some changes. Now, the other side of this is we've got the surface salt intrusion in the rivers and the estuaries. We've got the groundwater, but we are also looking at ways to mitigate. And we have locks, such as the locks at IJmuiden. We have the gates, the Haringvliet gates. But our coast is also subject to continuous development. And what we really need to understand is how do all of these different changes, these designs to our delta, impact salt intrusion, and how do we work with that and find the best solutions. So one of the ideas that we have, particularly in the project that Marianne was talking about, is to look at high resolution climate simulation. So this is a contribution by Pierre Siebensma. And what you see over here is basically the grids with increasing resolution towards Holland, so the atmospheric grids. And they can do these very accurate or high resolution, I should say, extreme weather climate forecasts. So you can see this here. And they can change the land usage and see what the impact of that is. So what we would like to do together is to connect the salt intrusion in the delta, in the groundwater, with the high resolution climate modeling. So salt intrusion is a national problem, as I have mentioned. We've got the drinking water supply for 5 million people. We have fresh water nature conservation areas in salt-prone regions, and we have a 10 billion horticulture and flower culture industry. And on top of that, our deltas are the entrance to fish and a hop for migratory birds. So we want to understand anthropogenic changes, and I really like this image here. This is by Marjolein Hassenoot and Jaap Kudijk at Deltaros. It's courtesy of, it's via Jaap. And what you see here are a number of solutions. What solution are we going to have? Are we going to have a fortress? Are we going to have an open system? And if so, what are the consequences? And these are the type of things we're trying to look at. And we've also got here, if we look at the Rotterdam area, can we change this region? Can we introduce building with nature solutions? Can we redesign it so that we can mitigate salt intrusion into our deltas far more cleverly? We've got building with nature solutions. Sorry, this example from New York. And I like this image by Art Natuurontwikkeling because it shows their vision on a healthy, open system. And that's really what we want. So we're trying to figure out how best we can do this and all the implications of these choices. Back to this slide, we know that this is happening in deltas worldwide. And if we look at salt intrusion in the future, we want to look at designing our deltas and designing scenarios for future land use, water and urban, as well as simulating future climate scenarios for the Dutch Delta so that we can understand better salt intrusion through our delta. And finally, some of the questions we would like to answer are, how will salt intrusion increase in the future? How will it depend on the design of the future delta? How will scenarios of future land use, water and urban impact salt intrusion in the delta? And how will our choices for the future delta impact the spread of viruses in the delta? So thank you very much. I'd just like to give credits to a lot of the work I presented is based on salty solutions. Many of my partners are listed here, and then also PSC, so Tom Bogot and the One Health Pact. Thank you very much.
So maybe introducing the next speaker.
Oh, sorry. Yes, I'd like to hand over to Martin. I'm very sorry. Hi, Martin. Yes, the next talk is going to be presented by Martin Schramer and Sander Kuhnrath. Thank you.
Martin, you're on mute.
Yeah, I know. Thanks, Julie, for quickly introducing me. And I think it's a very nice also introduction to the problem of what we're talking about today. This salt intrusion, besides all the ramifications for agriculture, for how we plan our landscape. I think this is something that a lot of people are interested in. But today, I think we really also want to go into how this impacts on diseases and disease risk. And what I try and convey in this very short talk is that these salinization issues that Julie just talked about have important consequences potentially for mosquitoes and their predators. So if we just show one of those pictures again, I think it's very clear that if we look at the Netherlands, we see that the coastal region, actually that urban delta that we are talking about here, is most vulnerable to these salinization issues. And that's also where most of the people live. And at the moment, this is a region that is about 7 kilometers wide, where most of the issues take place, and it's forecasted that maybe in the future it will be 30 kilometers wide where those changes will take place. Now, how will this impact on these diseases then? I think there's three major issues that we need to understand. First of them is, how does it impact on mosquitoes, both on abundances and their species composition? The second one is, how does it impact on the predators of these mosquitoes? And the third one is that how does this impact on the vector competence? To what extent these mosquitoes are able to transmit diseases? And this very shortly I will introduce. So the first one is actually about effects on the population dynamics of these mosquitoes. So from literature, if you dive into literature, you would see that we find that most of the mosquito species that we have here, for instance, Culex pipiens, are not able to deal with salt conditions. But we did a couple of pilot experiments in the last year, and what we see is actually this is not true. We find that the larval mortality is almost completely similar to if you grow them up in fresh waters compared to brackish waters, and we don't know what would happen in salt waters because we never anticipated that this would be the result, but it actually shows that mosquitoes are very able to withstand these brackish waters. You don't get more mosquitoes necessarily, but they can live in these environments. And why this is important, I will go into the next slide. The second thing is, of course, then you wonder, do they actually choose these environments? So on the right side of this slide, you see what would we call a cafeteria experiment. So places where mosquitoes can deposit their eggs. And if you give them different conditions in which they deposit their eggs, they seem to not make a preference for either fresh or broad brackish conditions. And on the right side, you see these egg rafts. And these are typically pockets of eggs of about 200 to 300 eggs each. So you can see this can be the harvest of a single night. So you can see what exponential growth, and I think since Corona, we all know very, very well what exponential growth is. You can understand how exponential growth can work in such environments. What we also see is that if you go completely salt, then mosquitoes don't like this environment to lay their eggs. But we see that these brackish waters can be great environments for some species. And this is done, as I said, with Culex pipiens, but you can also do it with Anopheles species, the species that used to transmit malaria, and we expect to find very similar results. And from anecdotal pilot data from the Caribbean, we know that some of the invasive mosquito species may have very similar behavior. So all in all, it seems that these salt environments may be very good actually habitats for mosquitoes. Now, the real problem comes in when you go and look at their predators. And this is a little graph of the dragonflies and their tolerance. And this is not actual tolerance done in an experimental setting, but this is just a graph with the maximum concentrations of salt where different species have been found. And you see that for dragonflies, and these are known predators of mosquito larvae, most of the dragonflies have lower tolerances. And this is only the top 10 or so that I plotted here. Almost all dragonfly species have much lower tolerances than the mosquito species, and that's maybe a real worry, because salt environments may create sort of predator-free environments and potentially hotspots in the landscape for mosquito growth, if indeed we start having these places everywhere on the coast. So that's something I think to think about, but also much more experimental work on this is needed. One reason to expect why these predators cannot adapt so fast to such environments is because the generation time of these predators is usually a year, whereas for mosquito species that can have easily 4 or 5 or 6 generations in a year. So their adaptive capacity, as we call it, is much higher. So so what I've got to tell you here is that the red one and the yellow one are the mosquito species, and all the blue ones are the dragonflies. And this is not just true for the dragonflies. We saw very similar patterns for beetles, for boatsmen, and actually for amphibians. They are known to be the best predators of mosquito larvae. They're completely not resistant to salt waters. Now, the third big unknown is whether animals, mosquitoes that grow up under salt conditions, are able to transmit viruses in the same way. Are they better able to transmit viruses? Are they worse in transmitting viruses? And that's a big unknown. And this is something that we can study, and we've done so before. And then when I say we, I mean the people in the University of Wageningen in the BSL-3 lab of the entomology group, headed by Sander Groenraad and Gorben Peilman. And if you grow up, so on the left side, you see the buckets where you can have different scenarios with different salt levels and different climate conditions. If you grow them under different conditions, then of course, then afterwards you can find out if these animals can actually transmit viruses by doing a very complicated procedure with blood feeding, and then trying to find out if actually the virus can also come out of the mosquitoes again. This is very specialistic work, but I think also very important to evaluate this question. And based on these measurements, you can have an idea about the infection rate, transmission rate, and then combining that with models on what happens with the temperature, what happens to the salt content of the water, you can find out what happens with transmission rates of various pathogens, of course. One big unknown in all of this is the birds. And Judy, after me, will go into that because the birds will be bringing in many of the viruses that we are talking about here. Now, this is all to understand the situation as we have it now and also in the near future. But to also understand what has happened in the past will help us a lot with what will happen, I think. And for that, we will also have an idea. We will try and use the historical records that are present on mosquito data, salt concentrations, and also climatic concentrations from the 19th century and early 20th century when we had a lot of malaria outbreaks. And you see a little poem on the left in Dutch. This was hanging in every doctor's room in the Netherlands, especially in the north of the Netherlands, North Holland, where there was a massive campaign to try and eradicate malaria in those days. And what you see in this picture, and I took this from a thesis that was written in 1902, this shows the north of Holland, and every place where there's a red line underneath, there was at least yearly occurrence of malaria in these areas. And this maps really nicely to a map of the Anopheles mosquitoes, so the mosquitoes that actually transmit this, and also to the salt concentrations of the waters in these areas. So you can here very clearly see already from just through your eyelashes how these salt water concentrations impact on, in this case, Anopheles mosquitoes, and then, in turn on malaria. And what I'm not saying here is that malaria is going to come back, because there's many more reasons why malaria disappeared than purely salt water content. But we can learn a lot from the dynamics of these environmental factors and the disease incidence on what might happen in the near future with other pathogens. So the five important questions that I think we'll have and we'll try and answer in the coming years is, first, how will a mosquito abundance be affected and which species will win where? Because I think that's important to see what's going to happen in the landscape. Does desalinization lead to predator-free environments? Does it lead to hotspots in the landscape? How does it impact on vector competence? Do these mosquitoes transmit the viruses better or worse? If it's worse, then maybe there won't be any outbreaks. If it's a lot better, then maybe we'll be in trouble. And then, of course, what can we learn from historical records? And the last but not least, and I think that is also a very important question that we always have, is it, to what extent are mosquito numbers the decisive factor? And is it maybe the most difficult part of the chain to break? Maybe there's other things that we can do. And for that, to understand that, I think we also need to collaborate with the other parts of the project, because that is a really important question. So with all that, I think it's very clear that it's important to understand things about mosquitoes, but it's also very important to understand things about birds. So that's where Judy comes in. So Judy, I will try and give the floor to you.
Thanks a lot, Martin. That was a great introduction for me. Let me just get my part started for everybody. And I hope I can explain to you in a very short period of time why we think birds are so exciting as linking pins in this story. So first, this is also very much from what we call an animal movement perspective. So if we consider the mobility of birds, birds are highly mobile. We can study them from many different perspectives, from the perspective of an organism itself, where it goes, why it goes there, what drives an individual animal to move to certain locations. Can consider movement from the perspective of the environment itself. How are environmental conditions influencing the movement patterns of birds? We do this with different measurement techniques, and I'll show you just some examples to give you an idea of what's actually possible. So we use things like GPS tracking, so small tags that are put on the back of birds where we can collect a lot of information about what they do. other things like radar to monitor how many birds are moving through the landscape. We use this fundamental understanding of avian movement to then try and solve different types of conflicts. And until now, our group at least has been working on a range of conflicts related to aviation, wind energy, changes in resources, but even conflicts with humans. And now we hope that we can also start moving towards the direction of trying to understand the role birds may play in disease, also considering the changes that we've heard about now and we expect going on in the Netherlands. So why birds? And what are we talking about as far as movements? Well, birds can create connections between very diverse biomes. They can do this on huge scales within their annual cycle. So if you consider the migratory movements, this is just an example from one species breeding in one colony in the Netherlands, lesser blackback gulls, that spend the summer in the Netherlands, and then they migrate out to a whole range of places, Western Europe, Southern Europe, but even all the way down to West Africa. So you can imagine that as these birds move to different areas throughout the year, they're going to interact and meet up with different bird populations and communities, different insect populations and communities, potentially bringing back moving diseases and other materials around during the year. And the Netherlands is particularly exciting and interesting for many reasons. First of all, again, another example from different types of species, waders, also wetland water species. The Netherlands is a hub for a lot of migratory species that move through the country and spend time here, again, at different times of the year. We've got birds that spend the summer here and breed here, other birds that will spend the winter here, but spend the summer in the high Arctic, birds that just pass through during migration, for example, in our deltas or in the Wadden Sea. So if you try and imagine these complex systems, which are changing and shifting through space and time, and try and imagine how complicated these connections are, but how important they are. And in the Netherlands in particular, we're also very well known among these movement flyways, these patterns, because of the wetlands that we have here in the Netherlands. And because they can support large numbers of birds, large densities at different times of year. What we also see at the same time is that we have populations which are changing. They're changing in their dynamics, the numbers of birds that we see here, either breeding or in the winter. And because the Netherlands is so densely populated, we think it's also extremely interaction because we can expect more interactions between wildlife and human populations because we are so close together. And there I want to make a step a little bit also to the link to urbanization. So our wetlands are very, very close to urban areas, sometimes even interconnected, or we have close transition zones. And something that we need to keep in mind is because these birds are so mobile during their life cycle, they are easily moving, or some species at least are moving in between these areas, often between the wetlands that we even have in city areas. So the species that we have in and around the cities will also influence what we may expect to see inside our cities. And here we can try and really study how these species connect these different environments. So just to give you some examples to make this a little bit more concrete from some of the work we've done before, here you can see an animation of birds, 13 individuals tracked during one month, June, while they're breeding in the Netherlands. And you can see how wide their actually distribution range is just during the summer. This isn't even migration. How you have individuals foraging very close to their colony, but some that go in and out of cities 70 kilometers away to Amsterdam, to Hilversum, coming back to Texel. Again, to give you an idea of what these linkages in space and time can be and how big these networks can actually be. And again, if we look at, to make this link again between different types of biomes, different types of environments, here you can see an example just of tracks. So the little dots are actually measurements every 5 minutes. We can see again how these birds breeding in an intertidal area, in a coastal area, will make movements every few days all the way to Amsterdam, 70 kilometers away. So we need an understanding of how far birds can move, how often they make these connections between distant environments, to also understand how they may be moving other materials such as diseases with them. Here's just another example of how cities can also attract birds from other locations. So this is another breeding colony in IJmuiden, and we know that waste treatment can be really attractive for certain species of birds, but even just simple human activities like people feeding them on a very regular basis. Something else that's also happening in these areas, which is intriguing, is because the food resources can be so high, it can attract very large numbers of birds for a very short period of time. So you get large aggregations of tens, even 100 individuals at once. They're there for a very short period of time. They disperse again to their colonies 20, 50, 70 kilometers away. So we get these high concentrations, interactions, dispersal again, and different groups meeting up and dispersing. So how does this link back to our research questions? Well, now if we consider what's going to happen with water management, with changes in wetlands, salinization, areas that are attractive to birds, and we consider the link to urban environments, what we'd like to try and understand is, okay, which species, and these are similar to some of the questions Martin has, but now from the perspective of birds, which species will be the most impacted by these changes in wetland management in the Netherlands in the coming years? We want to understand then how these wetland developments will also influence the avian communities. Which species are going to be in these areas, and how are they going to move? How are they going to create these connections that I just showed? And if we take a step further and we think about humans and human distributions, how are these connectivity networks also going to link urban areas with the surrounding wetlands? And of course, hopefully, the next step will be what is then the potential impact on disease and human risk? And this is also where Raina Sikema comes in, because she's also going to explain how we can actually measure the risk for humans and disease.
Thanks a lot, Judy. Let me share my screen. Yeah, so my name is Raina Sikema. I work at the Virus Science Department at the Erasmus MC, and I would like to go into the actual integrated sampling to assess vector-borne disease risk and the actual viruses that we monitor and investigate a bit further. So just zooming out a little bit, so why this important topic? So we know already that in the past years we saw an increase in different vector-borne diseases, which is likely due to climate change. This is partly due to the changed and increased distribution of vectors, but also vector survival, for example, overwintering of mosquito, but also, as mentioned before, is that temperature and other factors may also increase virus replication and the time frame in which this can happen during the year. At the right, you actually see the tiger mosquito and the projected changes there, an important mosquito to transmit several vector-borne viruses, and that is also projected to be much more widely spread throughout the world, with also larger periods of time in which they can actually transmit viruses. However, and that has been alluded to before also, is not only the climate change itself, but also climate change mitigation measures can be very important in terms of influencing and affecting risks and transmission of vector-borne diseases. So here I would like to go into one example, an important example for the Netherlands and for Europe, to show what we have already been doing, and to show also what we can do in the future here. And that is West Nile virus. West Nile virus was first isolated in 1940 in the African continent. Also, there were several separate outbreaks there, but it became actually quite a famous pathogen after its introduction in the USA, where we saw how such an introduction in a new area can actually exhibit itself. We saw massive bird mortality, east to west spread in 3 years, and also human and horse pathology and mortality. Since this is a pathogen in which a large part, of around 80%, exhibits itself as asymptomatic, but 10 to 20% we see flu-like symptoms, but in 1% this is a pathogen that can give severe neuroinvasive disease. And as opposed to some of the other vector-borne diseases that were mentioned before, such as dengue fever, this can actually be transmitted by the common house mosquito that is already in the Netherlands. At the right, you actually see a small patch in the Netherlands. So we detected this virus for the first time in 2020, and that was preceded by increasing spread throughout Europe. So just to explain again and to go back to the basics of why we are looking into birds and mosquitoes is that we actually know that birds are amplifying hosts, for example, of West Nile virus and other viruses. So that means that without birds, there is no amplification of West Nile virus, and transmission between birds and humans or horses is incidental, and also human-to-human transmission cannot occur. So these birds are really essential for the epidemiology of these viruses. And that is why, and this is an overview with a lot of different factors, but that is why we already set up in the Netherlands, and we aim to expand that also in the Rotterdam region, a system to try and monitor the actual introduction and spread of emerging viruses, not only in humans, but especially using birds and mosquitoes. So the first part, of course, is introduction. So we want to know when a virus is introduced. And in order to do that, we monitor migrating birds. They're caught and sampled, and we test for example, West Nile virus. And these migrating birds, we select species of which we know that they're susceptible, 1, and 2, that they represent different flight routes and different overwintering areas where we know what viruses actually occur there. Secondly, as a basis of this surveillance system is to monitor resident birds and domestic poultry, but also mosquitoes, which allows us to actually detect ongoing spread in the Netherlands, apart from introduction, and also allows us to detect where viruses may pop up and become a risk for the third phase, which is spillover, possibly to humans, which is obviously of high public health relevance. And we use different methods. So we can use serology, looking backwards at antibodies, has there been exposure and where, but also we use the virus detection and also trying by means of sequencing and further analysis, trying to determine the actual genetic code of viruses. And I will show how we use that information here. So, like I already mentioned, in 2020, we found the first West Nile case in the Netherlands. And this is the current monitoring and the current research setup as we have it now. So in blue, you see where in the Netherlands in 2020 we caught live wild birds and also monitored whether or not they were infected with West Nile virus. And the green dots and the black dots there, you see that we also monitor actual mortality, so dead birds, of which for some viruses at least they are better indicated as opposed to healthy alive wild birds. So the red crosses is where we also do additional mosquito monitoring. And indeed, in August 2020, we found the first common white throat, of which we knew was negative a few months ago, so pointing towards actual local transmission of the virus, and which was confirmed by also positive Culex mosquito pools in the same week, followed up by also in the same region, the Utrecht region, different bird species also being virus positive, including one chicken in a backyard. And this also shows the importance of why we do this, because followed up by this first introduction, which was special, it was the first introductions ever of West Nile virus. A retrospective screening was done in humans showing neurological symptoms with unknown cause. And then indeed, we found 8 human cases, also the first local human cases in the Netherlands. And this is maybe a complicated graph. So this is a phylogenetic tree. It can be read as a family tree. I'm just going to slide you to the side. And using this, so this is based on the genetic information that we got from both the mosquitoes and the birds. We can draw some conclusions or hypothesize a few scenarios in which we can learn on how actually viruses are spread and introduced into the Netherlands. So one of those conclusions maybe is that all of the sequences, we call them, so the DNA, the genetic code, are clustered together, which may point towards indeed local transmission and not separate introductions, maybe by different bird species. Secondly, we see that they cluster together with a very recent strain from Germany, which may, and there are caveats, of course, in all of these conclusions, but which may point towards the fact that that the virus is actually introduced from Germany. And thirdly, what we can also try and do is estimate when there was an introduction into the Netherlands. And that maybe mean that the virus was already around in the year before and that we detected it in 2020, although this has to be confirmed. So these are the type of analysis that we can do, and that how we at the moment try and find, for example, West Nile virus and other vector-borne diseases using as we call it, One Health surveillance system, so monitoring humans and animals, especially birds and mosquitoes. But ultimately, of course, and this I think is also a summary and a getting together of all of the previous talks, is we can detect and we can investigate different factors that we think are important, but can we actually predict vector-borne diseases? And this is a SketchUp. This is a schedule in which we also try to set up this front-runner project, and also the set of talks that was also in this webinar is how can climate and salt concentrations, changes thereof, affect the actual water management and changes thereof. How will that affect, and we think it will affect mosquitoes and birds, and we think it is actually likely that will affect human but also animal disease risk. And the question is ultimately if we can use this information to do one, more targeted surveillance, but two, also can we predict maybe these changes in disease risk, and can we also use this model outside of the Netherlands in other regions, as we saw, that have the same problems. And here you see those key questions again. So we are interested, of course, from the virus perspective, but ultimately we want to know, do indeed via these steps to changes in salt concentrations and water management indeed affect these disease risk for humans? And can we use all of the gathered data to predict occurrence of West Nile, but also of other vector-borne diseases? And with that, I think we can go to a discussion and maybe the questions.
Yes, thank you very much, all the speakers, for wonderful overviews. I think I get excited every time I hear the talks. So I think this is exactly the type of collaborations that we want to see happening in the PDPC. So I would like to introduce the other faces on the screen now. There's Professor Pearl Dijkstra. Maybe you can wave your hand. Yes, who is also a co-leader of the PDPC and who comes from a sociology perspective. And I'm sure she has some questions lined up. We have Sander Koenraad, who's not visible, who is visible now. Thank you, Sander. And who is co-leading one of the front-runner projects from Wageningen University with the great facilities that you saw presented by Maarten. There's Eldar Rakimbertiev. Did I do that well? Sorry. Yes, Eldar, wave your hand, from the UvA as well. And we have, oh, last but definitely not least, Piers Siebesma, who is actually the chair of this front-runner project and who has been trying to herd the cats and frogs into this wonderful collaboration, I think. So with that, let me open the floor for some questions. I would like to start with two questions that were in the chat. I know, Maarten, you have answered one, but I think it was an interesting question also for the broader audience. And that was a question by Lineke Begeman, who asked about, do you also consider other predators like bats, for instance, in the system? So let's hear what your answer was there.
Yeah, so actually, using we had a master student working on feces of bats last year to find out to what extent they're important. And Lineke was also involved in that project, and I thank you so much for your collaboration on that, Lineke. And what you see is indeed that some of the bat species are quite important, probably quite important bat predators. We still have to quantify some of that, but the bats are definitely interacting with mosquitoes, and maybe through that route also injecting pathogens. But besides active in feeding on mosquitoes, mosquitoes may be also feeding on bats, and as an intermediate transporter of viruses, bats may also be of interest. So I guess for me, the whole question about bats is a bit of a blind spot, but I'm wondering, maybe Judy, you know more about that. To what extent can we include bats in the kind of things that you showed, the radar based and
I saw your question. I saw your question in the answer to that question. I think for us, interestingly enough, for us, bat movements is also still something that is really in its infancy, especially because bats are, especially the bats we have in the Netherlands. In other countries with large fruit bats, it's easier, but bats are very small. Technically, they're very challenging to track at high resolution. So we don't really have the right technology yet at the moment to really do this fine scale measurements like I showed now, where you can really monitor where an animal is going during the day, how long it stays in different places. I think that we're getting closer. So there are technologies that people are using to study bat migration. That's something that could potentially be included. So I think there's certainly space to discuss this in the future, to think about how we can also integrate bats and maybe what's needed to take those steps.
Yeah, and I think just from what we saw in our preliminary research is that especially the ones in urban areas are very much feeding on mosquito.
Yeah, so maybe something to mention is to even think a bit, it's something we can think of out of the box a bit. There is quite a lot of work going on in urban areas to at least monitor bat populations, right, through acoustic monitoring and things like that. So there are things going on for different reasons for biodiversity, which may be interesting for this project. So yeah, it's a great question, a nice idea.
Yes, and Lineke Begeman was the coordinator of the Zoonosis in the Night project. And Lineke, I invite you to discuss this further with this group of people, because I do think joining up would really be good. I have two, I think, related questions for Reina, also from the chat. One is, someone wants to know, how do you actually sample birds? And there was a second question on someone understood that you do not do serology in birds, so it might be good to explain what you do exactly there.
Yes.
I ask everyone to be a little bit short in the answer so we can have a bit of a range of questions. Yeah.
Of course, yeah. So we do a lot, and it's of course difficult to all pull that together in 8 minutes. So first of all, how we sample birds is, of course, we have the dead bird monitoring. That is not too difficult, but the live bird monitoring, we work together with the bird ringing station in the Netherlands, where already birds are caught using mist nets mostly. And around 120 bird ringers in the Netherlands have had additional courses to be able to actually sample those birds. So that is why we can actually already also have those high numbers. So they sample the birds. We also draw blood and do serology. However, I haven't showed that here. So we also do that here.
Yes, thank you for clarifying that. And I may say there, I think there's some movies on the PDPC website, so you might want to check that if you are interested in this. Pearl, did you have a question? Did I see that?
Yes, I have a question, but first I want to say how wonderful this webinar is. At least I think so, because to me, it epitomizes the pandemic and the disaster. I see these crossovers. Another is I'm constantly thinking of policies. Right now, it's the farmers who want to keep their cattle versus the wetlands that bring all the birds to the Netherlands. So wonderful. I have one quick question for the first speaker, Julie. You had wonderful, the saline intrusion models, and towards the end of your slide, you said that was your third question, the spread of viruses, but I didn't hear you talk about viruses and the implications of your work, or how you're going to bring in the infectious diseases part. So maybe you can elaborate on that a bit.
Okay, thank you very much for the question and comment. So I'm absolutely not a specialist at all in viruses, but I'm learning here. And I think this is the great thing about what we're trying to do is cross-disciplinarity. So what we can understand is what might happen to the salt in the future, both in the groundwater and in the estuaries, and then link to the ecologists who can tell us how that might affect. For example, I think Martin's talked with the mosquitoes, also with the birds, the habitats. So I think I can't say exactly what would happen to the spread of the viruses. It does, if I listen to the connection to the other talks, suggest that salt intrusion in the future could have major consequences. And that's something that we'll hopefully be investigating together as a team. Does that answer where we are now? I was thinking short.
Yeah, no, I think that's exactly it. I mean, it is why this is such an interesting combination of expertise, because Julie is looking at landscaping solutions, but in one of our discussions, I remember you saying, "Oh my gosh, I've never thought about these."
No, I haven't.
And this is exactly why it is an interesting combination.
Because we really would like to maximize the building with nature solutions, but we don't know the consequences for the spread of viruses.
Exactly. And maybe in that line, I have a question. First of all, the quote for the day goes to Martin with your cafeteria experiments for mosquitoes. I just, a cafeteria will never look the same again. But my question was about this, both for you and Sander, about this imbalance of mosquitoes and predators. So I'm thinking of actionable information. So if you can see this coming, and of course that needs to be studied, but if you can see this coming, do you already have some ideas about how that could be influenced? Are we going to start culturing salt tolerant other predators, or are we going to flush our wetlands with
well, the latter way, that's how we do it at the moment. So we keep it fresh by flushing with fresh water, but that's a problem in the future because there won't be enough fresh water to flush everything all the time, particularly not in dry summers. So from species distribution modeling, if you know the predators of mosquitoes that are potent and they can disperse in the landscape, then you can also then you can know also how many of these places you need in the landscape to have sort of a background predation pressure present in your landscape, if you know something about the dispersal distances of these animals. And that's also why the collaboration with Naturalis is really important, because that's where the knowledge base regarding the distributions of these predators is present, and also how far these predators. I'm not a specialist in this, but if you have a landscape that is the Netherlands is very flat, but it's not completely flat. But if you have the landscape like the Flevopolder, where you have vast areas with nothing else than one type of land use, then you may run into bigger problems than when you have more of a mosaic, where colonization and extinction are balancing out. But I guess that is a very general answer to your question. I think in this case, we have to see how to go about that.
Okay, thank you. And maybe Sander, so you have been observing mosquitoes for all of your career. You study mosquitoes also in a tropical context. I can see people that are somewhat skeptical, saying, okay, well, this is long distant future, why worry? We have bigger problems to deal with. What would you say there?
Well, an interesting question, because indeed, as you rightly say, the focus right now is very much on the Dutch situation, the Dutch Delta, although we do include the various other areas that we say, like, okay, we maybe want to compare to the, like, how the other systems work. And I always find it important to keep a close link to things that we currently do in tropical areas. I mean, we've seen examples, of course, Zika virus, a recent one, chikungunya as well, that sort of have their origin in tropical areas, and that apparently have been circulating there for a long time without causing any problems. And it's something that triggers the sort of the sudden global spread. And I find it really intriguing to sort of find out, like, what does that trigger, and what does it make it spread and become global? And then indeed, sort of the question for us is also, like, to what extent is it really an issue and a risk for Northern Europe, so including the Netherlands. So I'm always in favor of indeed keeping close connections with studies that are ongoing in more tropical countries, well, to learn from what is happening there. And of course, that's also happening in the One Health Pact project already a little bit, and especially because at our bird migration patterns that make these connections, and it's understanding the interplay between sort of mosquito movement, bird movement, but also human movement that eventually determines our risk of new emerging vector-borne diseases. Okay. Yes. And the cafeteria experiment is indeed a sort of an existing term in ecology. So.
Okay. Yeah, that's really nice. I have a question, two more questions, because we are getting at the hour. One is about, so what we've learned from other emerging diseases is that, well, as Sander also just mentioned, you may see something percolated, but then something happens and things explode. So this is a question about abrupt changes that could happen and unexpected, difficult to predict changes. My question is about impact of, for instance, a very severe weather event on bird movements. I've seen examples of that in Curacao, where there was a bird sampling program, and then with hurricanes, you would all of a sudden see very different birds that were just blown out of direction, essentially, and were then in a new area. Is that something that is theoretical for the system that we are looking at here, or is that something we should also be thinking about? Maybe Eldar.
Thank you, Marion. Yeah, I think we have, in the Netherlands, we have birds coming from all over. So of course we have birds that come from the Arctic, from the American Arctic, for example. So if we have storms, we also get some juvenile birds blown here, for example, from the US. But at the same time, we have just normal populations coming here from Canadian high Arctic. So I think the connectedness that we already have is enough to assume that everything is connected. But it goes within, like they go up, then they can get infection, then they go down to the other place, right? And with this storm, you can get like horizontal transmission that is faster, but it's only faster. It's not that it increases the probability of connection. That's my feeling.
Okay, so it could be sparks.
Yep.
Okay, last question, and that's a sort of questions from one of the co-leaders of the PDPC, Bas Jonkman, who has questions about what could be the most effective strategy to reduce the risk of diseases. So that's essentially a question for all of you. And there was another question. Oh, I think that has been answered. Let's stick to this question. So final question for all of you. If just out of the box in the future, what types of prevention measures could you think about? And I know we don't have the data. I know we will need the research, but just out of the box. So
if I may start. My feeling would be that having a strategy is maybe already a start. I think it links also to avian influenza discussions where we see that large poultry farms are still located next to large wetlands, for example. And in this case also we saw that discussions on salt intrusions are really very separate from discussions on disease risk, and very separate on how we actually develop our houses and cities. So I think that could be a start, and I'll keep it short, but I think a more integrated approach without, I think, having the possibility that there's one best answer would be good, and maybe to have a national strategy on where we would want certain functions and what we should combine.
Okay, great. Judy?
Yeah, I'm thinking maybe if I just follow up on Raina, it's a little bit easier, right? So if we think about things like these poultry farms, you can think about spatial planning, right? But you can also think about temporal planning. So I think if we also know quite a bit about the phenology, the timing of large movements of birds, for example, we can then think about how to structure in time when animals are out, when animals are indoors, so that you have a proactive approach. So these things we might be able to combine them. The same thing as far as interactions in different types of areas. Just as short as I can make it for now.
Great. Maarten, you already mentioned the flushing.
Yeah, and I think based on the data that we'll gather, we will be able to make a sort of prediction map of where mosquitoes are likely to pop up, and this will be areas where I guess you would like to make sure that at least the predation side of affairs is well organized. And there's many ways I think you can go about it, but you have to first, have some kind of prediction, and you have to acknowledge this.
Thank you. Julie?
Yes, I was listening and thinking about this. I mean, we can develop different scenarios for the design of our deltas, and that's what we've been discussing, whether it's in salty solutions or also with groundwater. And given those scenarios and looking at the effect of climate change on them, then we could give that back to the ecologists and virologists to say what could be those consequences. And I agree with what you were saying earlier, at least from what I've picked up on, that you might want to separate poultry farms from other, so say nature conservation areas where you might have more birds, if I understood properly. Thank you.
Okay, so and then finally, Pierre.
Yeah, well, this question actually illustrates very nicely the dilemma that we are also in, because if you think about risk decrease for diseases, I mean, you might want to have less room for rivers, right? Those scenarios. But on the other hand, I mean, that solutions we are planning to reduce the risk of extreme weather and rain. So you see that, and that's in a way what makes it difficult, but also what makes it so interesting that we have these groups having together to look at this conflicting type of threats that we are facing, and that we have to somehow to optimize the solutions, taking both into account.
Okay, thank you very much. I think this was a wonderful discussion. I hand over back to Fred for closing this webinar. Sorry for being a little bit over time.
No problem. Thanks everyone, all the participants, speakers and panelists for sharing this fascinating research. We hope to see you in the next PDPC webinar the coming month. Thanks. Have a nice day.